Um, Today, it's my pleasure to introduce Dr. Lai Guan Ng from Shanghai and deliver this MCC seminar this morning. Just a little bit background about Dr. Ng. Lai Guan grew up in Malaysia and he did his undergraduate research training at University of Sydney, Australia. And then he continued and obtained his PhD at University of South Wales, University of New South Wales. Sorry about that. And then after that, he did a few postdoc and research trainings. First at Institute of Molecular and Cell Biology at Singapore, and then the Vista Institute in Pennsylvania, and then the Centenary Institute of University of Sydney. And in 2009, he became independent and become a principal investigator at Singapore Immunology Network Singapore, and rose the ranks there. And last year, he moved to Shanghai and become a professor at Shanghai Jiaotong University and also a senior investigator at Shanghai Immune Therapy Institute of China. So for those who know Lai Guan, Dr. Ng has been always expert on neutrophils and monocyte development, and his group has utilized a state-of-art technology to really study neutrophil development, and recently also the function of neutrophils in cancer. His lab has made several profound findings in the field of neutrophils, and his work comes often to journals such as Science, JMAT, and Immunity. I met Lai Guan this summer at an international meeting. We had a couple of beers together and became friends. Besides a scientist, Lai Guan is a very fun person to get along with. So I'm really looking forward to your seminar, Lai Guan. Welcome to Minnesota. The stage is yours. I want to make sure about the light.
Light.
Better.
Okay.
I know you have a lot of images.
Okay, so thank you, Haiping, for the kind invitation to come here, because in fact, Minneapolis hold a special kind of a place in my heart because of actually at beginning of this year, I organized a Keystone meeting in Canada, and for some reason I was trying to board the plane to Canada. I wasn't allowed, so pretty much I missed my own meeting that in Canada that I couldn't attend, and I spent a few days here. But that time I still don't know Haiping yet, so I kind of I like this city quite a bit. So but some of my friends who attended the meeting is like they kind of knew that I got stuck here, and then I was sending them texts and messages to tell them I'm actually back in this city that I love so much these few days. So it's good to be back, and then also good to be able to interact with some of you guys here today. So the way I kind of structure my talk today is that I kind of put everything together in terms of what we have done in the past few years, or I should say since we started our lab in Singapore about 13 years ago. So I always use like either neutrophil one-trick leukocyte, or I use a neutrophil the power of many, and some of you guys may know Ido. Ido is always say the power of one, the title of his talk. But for me, I always think of neutrophil is like the power of many, because like you need a lot of them to get something done. And coming back to this is like neutrophil are the major circulating cell, as we all know, in our circulating system. And more importantly, like we also use neutrophil as a kind of indicator in terms of when you do a blood test, and then to look at whether there's an infection or inflammations by just looking at this neutrophil number or frequency. However, but for a long time, neutrophil kind of been considered as a pretty boring cell in terms of what they do. They are professional killer. They just like blindly chase down the bacteria or pathogens, and then they would engulf, eliminate them, and pretty much that's what they do, and short-lived, and also homogeneous populations. And because of that, it's always like, when I go to, for years ago, for many years, when I go to meeting, it's like, you don't get so much respect in that sense, or very difficult to get a talk even. And this is very clear if I show you this slide. When British Society for Immunology, when they organize this annual meeting, they have all these badges to give out to the trainees or to the participant. And luckily, Neutrophil is still in there. But one of my very close friend who did a PhD together with me at Garvan, when she talked about immune cell interaction, you can see she included fibroblasts, not even in neutrophil. So you can see that how underappreciated neutrophil has been, even though they are one of the the most abundant cell in our circulating system. So why do I with all these kind of things that I've been talking about, they have been underappreciated. Why did I even bother to study them? Because I started my postdoc in a immune imaging lab. So we use a lot of intraviolet imaging to look at different immune cell. And when I became independent, I thought, I want to do something that's still immune imaging, however, want to slightly different from what my former mentor was doing. So I came to a conclusion like I should look at the fastest moving cell, because if you want to look at something moving, if you look at for example, macrophages, which doesn't move, is kind of boring. So I choose to work on neutrophil just for a very simple reason. And because of that, as a lab, when we started, we established various intraviolet imaging model, including the bone marrow and skin model that we use quite often, that to look at, for example, you can see here that you can see like how this neutrophil been activated when you inject GCSF within the bone marrow, how this neutrophil been released into the circulations from the parenchyma of the bone marrow to the sinusoid, and then released into the circulations. And then we also look at how this we also define these a three-step cascade of recruitment of neutrophil, in which we call them scouting, amplifications, and stabilization, that we reported 2012 which is similar to perhaps if you've seen Ron presented, Ron also have these similar observations. And we also look at how immune complexes may trigger neutrophil responses. You can see this white signal here represent the immune complexes, and how these neutrophil can carry some of these immune complexes with them when they transmigrate and migrate into these interstitiums. So all this movie is kind of like, when for the first few years of my life, we really focus on intraviolet imaging, trying to define how this cell carry out their functions in vivo. And one of the, actually I should say, two of the major things that we realized at that time is that neutrophil works in large number and very rapidly. And all these movie that I show you is like within 20-30 minutes. So it's a large number, rapid. So then this kind of raises questions. As I mentioned before, they are relatively short-lived, 6 to 12 hours in the circulations. And which also means that if they're short-lived and you need so many of them so rapidly, so you have to generate a lots of them to meet the demand. Then this is kind of shifted our focus as a lab into asking where they're coming from. So we know that they are from granulocyte macrophages progenitors. However, in between the step are not so well defined. When I say that not so well defined, is referring to because I mean, in the past, classically, they can be defined based on their granule content or the nuclear morphology, and based on this, you can kind of define their developmental trajectory. However, there isn't a very defined surface marker or transcription factor that help you to really like pinpoint like this is the step that the neutrophil are at. And because of that kind of reason, we kind of started to perform whole series of experiment. And one of the major so-called like the hypothesis that we had at the time is that if we need so many of these neutrophil, which also means that the progenitors or precursor, they will need to be highly proliferative, so that you can give rise to enough mature cell at the end. So because of that, we use proliferation as a marker, hoping that we can use the proliferation as a marker to help us to identify the precursor. And with that, we make use of this CyTOF at the time in collaboration with Evans Newell when he was still with us in Singapore. and we label like the IDU, which kind of will mark the proliferating cell, and we combine it with 40 different other antibody. And with that, we then look at the IDU positive, which is proliferating cell, and then the IDU negative non-proliferating cell, and we try to search for the differentially expressed surface marker that can help us to identify the precursor. So with that, at that time, we identified this pre-new, we call it pre-new neutrophil precursor, which is CXCR4 intermediate and c-KIT intermediate. And with that, we can better kind of define the mature and immature neutrophil based on CXCR2 and CD101 that we also defined at that time, which is like CXCR2 positive or CD101 positive will be the mature cell, negative will be the immature cell, and the mature cell will be more resembling to this circulating mature neutrophil. And this paper is published a few years ago, so I won't go into detail. Essentially, when this paper came out, we have this kind of provided a framework for the community that they can use several markers that we identify to really isolate out each of these neutrophil subset for further study. So with that, and when Max graduated, he went to Australia for his postdoc, and then Emmanuel came along, and we have one of these things that initially we started is to hoping to use this proliferative capacity of this precursor cell in terms of treatment, using it as a treatment. Like a cell therapy treatment. What I meant is that the cell therapy treatment will be like we can use this precursor cell to co-transfer it together during the bone marrow transplantation, because as you may know, bone marrow transplantation will take 3 to 4 weeks before the neutrophil are reconstituted. So we thought that with this precursor cell, perhaps we can bridge the gap between this reconstitution period. And however, Emmanuel found that when we try to look at this cell, they can only divide 2 to 3 times, which doesn't really make sense, because if they have such a low proliferative capacity, it shouldn't give rise to enough cell that we need. So we kind of start to ask the question, could it be that how we define GMP all along, which is not 100% accurate? It could mean that this GMP populations already consists of populations of these committed neutrophil progenitors, which should have a higher proliferative capacity. So that with that in mind, we then make use of this screening method, because now that we know using the proliferation may not be accurate, 100% accurate. So we should go even further up. But at that time, we don't know how to separate out the GMP population. So with that, we make use of this infinity flow workflow designed by, I should say, established by Evans Newer and Florentino when they are both in Singapore. So essentially, what we did here is that we stain this bone marrow cell with the 13 backbone marker. In our case, we stain it with all the neutrophil related marker. And then we leave the PE channel empty, and then we bought this BioLegend kit, which consists of 255 PE conjugated antibodies, and then we screen through one by one and collect this data by flow analysis. And then with that, we can use this algorithm that they design to compile all these flow data sets, like 268 of it, 255 of it, into one big single FCS file, and then we can kind of do a further analysis. So long story short, essentially we can kind of project all these things into the UMAP space, and the key is that as I mentioned before, we know that the neutrophil is from GMP, and then with this screening, essentially we kind of close the gap or bridge the gap from this GMP or to the pre-new. So we identify 2 progenitors population, pro-new 1 and 2, which is like CD can be further like separated into CD by CD106, which is like pro-new 2 106 positive, pro-new 1 is 106 negative. And then this study is published in 2020, and essentially by doing this kind of very systematic way, from starting from a proliferative capacity with combined with this kind of screening, we then essentially provide a whole trajectory like with a defined marker that now we can use to study mouse bone marrow neutrophil development in the bone marrow. So you may ask me, what is the use of defining such such detail? So I hope that through this slide, you will appreciate that by having all these marker, it can actually help us to understand the biology even better. So for a long time, CBP epsilon knockout mouse has been known that to be devoid of this mature neutrophil. However, at which step this development is arrested is not known. So using all these markers that we already have, you can see that without this transcription factor, essentially the neutrophil kind of got blocked. The development is blocked at this ProNeu1 stage. And somehow this will give rise to this atypical granulocyte, which is CGLF positive and CD1115 positive. And then this is not present in this wild type mouse. So essentially, by having this very defined marker, it will help you to better understand, how this neutrophil develop in terms of imagine if you found another transcription factor, you can essentially use this system to look at which step this neutrophil development is blocked. So the other thing that we then have to prove to the reviewer at the time is that we have to show that all this using this marker, we can robustly identify such precursor or progenitors, and they are the committed progenitor. It's not like a mixed population that we have. So we sorted ProNeu 1, 2, and pre-new, essentially using our markers, and you can imagine that we sorted them from 3 different color mice. GFP, and then 45.2, and then RFP mice. So it's like a 3 color and 3 different stages of development, and then we transfer them back into a recipient, a common recipient, so that they will be like kind of in the same environment. And after 3 days, we collected their bone marrow and look at their where they are at in terms of their development. And what you can see is that with this ProNeu1, so essentially after 3 days, most of it is in an immature stage, which is CXCL2 low. And then with this one step further, these are ProNeu2, you can see there's about 30% of it is becoming mature neutrophil, with still majority of it are still immature. And then pre-new is the stage that the closest to the post-mitotic stage. You can see that most of it is already becoming a mature neutrophil. And more importantly, if you look at all these 3 stages that are so-called the progenitors that putative progenitors that we isolated, they do not give rise to other cell, for example, monocyte or eosinophil. So it showed that using these several markers that we identify, we can isolate these committed progenitors robustly. So with that, as I mentioned before, this CD101, we also identify this key marker, especially in the mouse system. For a long time, how the field has been kind of defining the mature neutrophil versus the immature neutrophil is based on Lys6G and CD11B, slightly lower. Then they say that, okay, this is the immature neutrophil. However, with this CD101, we can now more robustly to show that like with the mature neutrophil will be definitely a CD101 positive population. Immature neutrophil will be the CD101 negative population. And using this marker that we discover, we can now say that in the blood at resting state, you only have this mature neutrophil in the circulation as well in some of this tissue, even at resting state. And under stress condition, for example, injury or tumor, tumor-bearing mouse, you can see that both the mature and immature cell can be detected in the blood, and both of them can be also recruited into this tissue as well. Oops, sorry. So with that, we also try to kind of because since all these in the mouse system, we want to kind of look at it, look at whether we can replicate all these in human. So using the same approach, we then perform our screening in human cord blood, and using the legend screen and infinity flow approach, essentially we can again define this neutrophil from ProNeu all the way to the mature neutrophil. However, I have to admit that, like with these studies that we have done, I don't think we have done such a detailed job as these three other studies. For instance, the immunity paper by Lin, and also by Marco, as well as Renato, that they really look at it in a much detailed way. If you are interested, you should refer to their paper as well. So coming back to so with all these things, what does it all mean now for the community? So essentially, we kind of put all these framework out there for people to use, and together with the surface marker that we define. But if you look at it closely, if you look at different lab, essentially we are looking at the same thing, and depending on the so-called the resolutions that you are looking at, we are looking at the same trajectory, same continuum of neutrophil development, but somehow we kind of call it a different name, which is kind of not so good in terms of like unifying the field and making it easier for the postdoc or for the student that who are not really working on neutrophil. This is something that we are still trying to somehow get together. We have this meeting several times, but we still cannot have a consensus how to put them properly. It's something that we need to do better in our field compared to the monocyte and dendritic cell field, which they have done a good job as. And because of that, I think it helped the field to grow even bigger, because it's much easier for people that do not really work on the field to come in and to work on this topic. So with that, as we put out all these things, I have a habit. I don't know whether it's a good or bad habit, which is to check who cite our paper. So I have this habit checking, like who's citing our paper. And initially we thought that perhaps the hematology field or the so-called the developmental field will pay more attention to what we do. But to our surprise at that time is that in fact, it's the tumor immunology field that kind of pay attention to what we do, because they want to use some of the framework that we propose to try to understand our neutrophil in tumor. So these are the several review articles that kind of cited our paper, as well as recently there is this JEM that they try to so-called like put a consensus statement. If you read it carefully, there's still no consensus, unfortunately. But in any case, they kind of cited really heavily all the markers that are both in human and mouse that we identify, and trying to put this in the context of tumor. So with that in mind, we thought that instead of letting other people works on what we kind of put out, so we should work on it ourselves, and that is kind of taking to the next phase of what we have been doing in the past about 4 years from when we started all these projects. So we started to ask this question, like, what are tumor neutrophil? Because it is I cannot say it's totally unknown. It is known. However, it's a bit confusing in terms of how they are called, because they can be called N1, N2, MDSC, myeloid-derived suppressor cell, or tumor-associated neutrophil. And all these naming system, if you look at it closely, is based on their function. There is no specific surface marker or specific transcription factor saying that these are the tumor neutrophil that you're looking at. And the other thing is also neutrophil roles in tumor genesis or tumor development has been reported can be both anti-tumoral or pro-tumoral. So to us, it's kind of a bit confusing, like the whole thing. So we thought that perhaps we can start from some of these framework that we already established, some of this expertise or workflow that we established. Can we kind of use this to further kind of understand it better, and hopefully we can better characterize what they truly are in terms of their role, their functions, and how should we look at neutrophil in tumor. So the three questions that at that time we wanted to address is that we wanted to know whether this is a tumor-induced state, whether this neutrophil somehow during the tumor development or tumor genesis, they kind of went into, they kind of have this a very unique kind of functional state, or actually there's a preferentially recruitment of a certain subset of neutrophil into the tumor microenvironment, or in response to tumor development, bone marrow or extramedullary hematopoiesis in the spleen, they will generate a new subset of neutrophil. So these are the questions that we thought that if we can better address it, perhaps it will help us to better understand what are the roles of neutrophil in tumors. So to address that, we make use of this KPC orthotopic model, which is a pancreatic tumor model. And what we did at that time is that we implanted this tumor into the pancreas, and for 6 weeks, and then we isolated neutrophil from all these tissue or organ, including the tumor tissue, and we perform hashtag analysis by 10x, and then and because we can't combine this into one experiment, so we don't have to go through any kind of so-called normalizations. We can just project this data set into the UMAP space, and what you can see immediately is that how these cell cluster very nicely according to their developmental stages, from the precursor immature to mature neutrophil. These cells are from the bone marrow, blood, and spleen. Whereas the tumor neutrophil from the tumor tissue essentially just cluster on top of here. So with that, we then wanted to kind of validate our data, because I have to be honest, like, when we started these studies, we are not so familiar with the technology at the time. And then we kind of make use of these studies that are already published in 2020. I would say it's one of the very first data set that published out there for the community is from Hongbo at Harvard. So they put out these single cell data sets from mice, from the bone marrow, spleen, and blood at the resting state. And then using their data, we kind of integrated our data. And then you can see that in the bone marrow data set, by integrating our data, you essentially you don't see any emergence of new cluster or new branches. So it's kind of suggesting that perhaps that the bone marrow do not generate a new subset of neutrophil in response to this tumor development. However, we did observe that there's this expansions of the precursor as well as the immature neutrophil population or the cluster in this spleen of this tumor-bearing mice. But if you look at the trajectory closely, it resembles very closely to this bone marrow trajectory. So it kind of suggesting that perhaps there is this increased EMH, extramedullary hematopoiesis. However, there's no deviations in terms of their development generating a new subset of neutrophil. The only thing that we saw that is very distinct from their data set is our tumor data set that is not found in their control mouse data set. So with that, Melissa then kind of further subdivide them into 3 kind of smaller cluster, and we had this debate for a long time. Should we try to name it more fancy, like instead of just calling them very boring, like tumor neutrophil 1, 2, and 3. So but in the end, we decided to stick with simple stuff is easier. So we call it T1, T2, and T3. And the very first thing that we look at is the maturation score, because our rationale is that if we can look at their so-called the status of their maturations, if these cells are less mature, most likely they will be more like a newly recruited to the tumor tissue. So when we look at it, what we found is that quite interestingly, the T1 represent more of these immature cell, T2 are the mature cell, T3 are kind of a mixed populations. So with that, it kind of it becoming quite interesting for us at the time. So in order just to kind of double check our data, Melissa then also kind of put it in I should say analyze it in a different way using this diffusion map. Again, the trajectory can be clustered very nicely, and then the tumor tissue neutrophil will be just clustered at the bottom here, and when she kind of integrated this with the RNA velocity analysis with this direction of this arrow is kind of inferring how they develop. We can see kind of a very nice trajectory from prenatal to immature to mature neutrophil, which is what we know. But more importantly, what she found is that these T1 can come from this immature cell, and then T2 is coming from this mature cell. And then if you put everything together, you can see that kind of T1, T2 somehow converge at this T3 population, which is a mixed populations. Then this is kind of raised questions that we, from our flow cytometry analysis, we know that both the mature and immature cell can be recruited to the tumor tissue, which could represent the T1 and T2. And then the question at the time for us was that whether these so-called the immature cell, they have to go through this maturation process before they can be reprogrammed into this T3 neutrophil. So that was our questions. So to address that, we make use of this CD101 marker that we discovered. We isolated the mature neutrophil, immature neutrophil from tumor tissue and various tissue and organ from the tumor-bearing mouse and control mice. And then and perform this bulk ataxic analysis. And what you can see here is that with this so-called analysis, you can see that only the neutrophil, regardless of their maturation status, we can detect this open chromatin region, but not in other tissue or organ. So it kind of suggesting that perhaps the predominant reprogramming is occurring within the tumor microenvironment. And then this is kind of the summary slide. And I always use this slide to kind of explain this concept because it will be much easier to understand us. Perhaps some of you guys will know Florent Ginu, a very good friend of mine. We started together around the same time in Singapore. We are very two different person. I always think that I'm more mature. He's more immature because I always do the right thing. We are good friends, so I'm just joking. And then we are kind of a very different intrinsically. However, when we go to the same microenvironment, which is like in this case here, we have this outing from our former institute that we went to the Singapore Zoo. So we put on the same uniform or same T-shirt. We are doing the same function. So which is like kind of looking at different animals at that time. So you can also imagine like, we can be two different person. We put on a lab coat and then we start doing the same experiment. So regardless of intrinsically how different we are. So with that, now that we know there's T1, T2, and T3, then we started to ask the questions. What does it mean? Like, do they these three population, do they kind of localize together, or they're distinctly kind of distributed within this tumor microenvironment? Because by having this information, it will then kind of further consolidate or further support, our observation that there are three different kind of subsets of or subpopulation of neutrophil within the tumor microenvironment. So we perform this spatial transcriptomic analysis using this 10x Visium approach. And then the take-home message is that what we found is that the T3 neutrophil essentially co-localized with these high glycolytic, hypoxic, and angiogenic regions. And then so far, with all the things that I've been showing and sharing with you, is that we have been kind of doing this study based on a transcriptional profiling, and as we all know that transcription may not mean anything if there's no protein. So we always want to go back to the protein level. So for us, the best will be, if we can find some even like a surface marker, because with that we can further study them even better, as well as perhaps this marker or this surface molecule can be used as a therapeutic targets. So again, we make use of this infinity flow and legend screen approach. I won't go into details because I mentioned this approach already in the previous slide. So essentially, we screen through this tumor tissue neutrophil, and however, we set these two important requirements. So we want to find a marker or markers, a series of markers that only expressed in the tumor neutrophil, but not on other infiltrating leukocyte. And more importantly, we want this marker that can only be induced by the tumor microenvironment, but not due to any other associated informations. So with that, with these two requirements or criteria that we set, we perform this screening, and you can see that this autotopic PDAC model is a very neutrophil-centric model, which consists of a lot of infiltrating neutrophil. And long story short, essentially, we found one marker which fit into this two requirement that we set for ourselves, which is this DC3R1. And with that, we found that this DC3R1 specifically enriched in this population here, but not on this big population down here. So we kind of hypothesized that perhaps this DC3R1 positive neutrophil can represent the T3 in our single cell data set. And with that in mind, we then went back to our single cell data set and we look at the gene set, gene expressions, and we also found that the DC3R1 indeed they are one of the actually the top 20 highest expressing genes in these T3 populations. And then with that, we kind of say, okay, perhaps this is the putative T3, and then which also means that this big population down here should consist of T1 and T2. And then with that in mind, we look at CD101. As I mentioned before, T1 is a so-called more like the less mature neutrophil, and indeed you can see that they express lower level of this CD101, and T2 will be this more mature one, high level of CD101, and more importantly, T3, as mentioned, it can consist of both mature and immature neutrophil. You can see that there's this one small population of this CD101 low immature neutrophil within these T3 populations. So with that, we then thought that, okay, so now we found these several markers. We have to validate whether this can be truly the marker that to help us to isolate these putative T1, T2, and T3 populations. So we sorted this cell based on this marker, and then we perform sequencing again, and then we map it back to our single cell data set, and you can see that it kind of mapped quite well, and kind of suggesting that this T1, T2, T3 can be identified at a protein level using these several markers that we found. And with that markers that we already we identify, we then can perform many more other study. So one of the first thing that we did at that time is that we want to validate our spatial transcriptomic data set. So we make use of this Maxima, Maxima platform from Mutani Biotech. Essentially, it's a cyclic imaging platform. You can imagine like every time you stain is a 3 different color or 3 different antibody, and then you will kind of erase or bleach the fluorochrome, and then you can come back in with another 3 antibodies. By doing this kind of a cyclic process, you can then stain for multiple color. So we have tried to stain up to 90 over different antibody, but in this case here, we stain for about 25 antibody to define these few things that we want to look at. First is the niche, and using these markers, we could kind of define several key niche that we want to investigate. And then I think this will be a much better image that you can see that like with this 73 will be, the low or intermediate, we will call it like kind of parenchyma, and then negative will be the stroma, and then with this 105 will be the vessel, and if this double positive or double staining double positive for 73 or group 1 will be hypoxic glycolytic region. And then since that we know how to stain T1, T2, and T3, we can co-stain this as well. And with that, we can then perform, I have one slide missing. So we can then perform this identification using the histocytometry methods that defined by Ron Germain lab to really extract the data from imaging and then analyze it like a flow cytometry data set. And then we kind of, I'm sorry that one side is missing. So essentially we show that consistent with our spatial transcriptomic data set, T3 is co-localizing with this hypoxic glycolytic region. So with that, we then kind of move on to show that now that we know that there is T1, T2, and T3, both transcriptional level and then also protein level, they are distributing very differently within the tumor microenvironment. So we wanted to kind of further understand their functions. So we sorted this T1, T2, and T3 neutrophil, and then we performed this co-injections kind of a model that we established. Essentially, we are co-injecting the tumor cell together with a different subset of sorted neutrophil, and we implanted sub-Q to the mouse, and then we monitor the tumor growth. and we boost every 3 days. We kind of boost it with a different sorted neutrophil again. So what you can see here, the T3 essentially is a pro-tumoral subset. It can promote tumor growth over time. And then whereas the T1, T2, and other control neutrophil subset, they don't have this ability. And at the end of the experiment, when we collected the tumor tissue, and then you can see that these T3 co-injected tumor are much bigger than the rest of the control groups. So with that, we asked ourselves what would be the possible mechanism that are promoting this tumor growth. So at the time, we kind of hypothesized that one of the very first thing that we hypothesized that perhaps T3 will have this pro-angiogenic kind of a function. So and then you promote blood vessel growth, and then this will then promote tumor growth. So we immediately we can then go back to look at our single cell data set, and you can see that indeed the T3 is enriched in these VGFA expressions. And the good thing about having both RNA kind of data set and protein data set is that you can kind of switch back and forth. So immediately we can jump to look at the protein level whether T3 will be expressing a high level of VGFA protein, and you can see that through our intracellular staining of this flow experiment, indeed T3 is a highest expressor of this VGFA protein in comparison to T1 and T2. So now that we know that the T1, T2, and T3, only T3 can promote tumor growth, and then they also express high level of VGFA, which is a pro-angiogenic factor, then we ask ourselves, can we really truly see the blood vessel growth within this tumor? And instead of using a 2D section approach, we thought that because blood vessel is a 3D kind of a structure, so it's better to study 3D structure with a 3D approach. So we make use of this platform that we established years ago when we were in Singapore. Essentially, it's an optical clearing approach in which we can take out the tumor and we optically cleared it, and then we stain with CD31 and we perform this 3D imaging and we quantify this blood vessels density. And you can see, as you can see from these videos here, the T3 neutrophil co-injected tumor, they have this increased blood vessel density network within these tumor core regions, whereas the wild type co-injected control group, the tumor is much smaller, and which coupled with this absence of this blood vessel growth within the tumor core regions. So this is a quantification, and you can see that the T3 co-injected tumor, they have this increased blood vessel volume at these regions. So this is a paper that just came out in January. So if you're interested, you can follow this link, and then you can have a more closer look, because there are many data that I did not show here. So essentially, with this, I hope to convince you that neutrophil they are not a homogeneous populations. So if you want to look at a neutrophil kind of a response, perhaps there's a multiple consideration that you should take into account. For example, like if there is a stress condition or stress kind of signals, you really have to look at it in a compartmentalized way to better understand them. For example, in the bone marrow, what's happening, and then also whether there is this premature release of this early release of this immature neutrophil, and then how when they are recruited to this effector site, how they may be reprogrammed. And in our case here, when these cell, regardless of their maturations, when they are recruited to this tumor microenvironment, they got reprogrammed into this T3 pro-tumoral T3 subpopulations that can promote tumor growth at least through this one factor VGFA, which promote the angiogenesis. So you may ask me, Lai Guan, you have been showing me all these neutrophil data tumor model is all in mice. What does it mean in terms of this real world, in terms of clinical applications? So I just want to share a little bit, touch up on just before our paper, actually is way before our paper came out, that this 2022, there's this paper, they look at this tumor, liver tumor especially, and then they already in human patient, they already reported, neutrophil heterogeneity. So that is one thing that kind of consistent between mouse and human, which is, we all think that there is a heterogeneity within this tumor microenvironment. And then this paper actually stressed me out quite a bit because we are trying to, wrap up the paper, and then this paper came out, human PDAC, and then look at neutrophil, which is very, very similar to what we do. But now we are very good friends because they are also in Shanghai, and at that time they are very kind to share their data with us so that we can also analyze their data in our paper. And some of these authors in this paper actually become a friend and co-author of our paper, which is a good thing about, when we collaborate nicely together. So what we did at that time is that we extracted their neutrophil data from this patient, and using our T1, T2, or T3 as a signature or reference, and reference signatures, and we then kind of projected to our kind of UMAP, extracting their data set. And you can see that for this tumor derived from this I should say neutrophil derived from this tumor tissue, they became this T3. They have this T3 signature. However, in the corresponding patient blood neutrophil, they do not have this T3 signature. So again, this kind of show that what we propose, neutrophil has been reprogrammed within the tumor microenvironment instead of when they are in other places. And more recently, there's this paper that came out in Cell, which, because our paper came out before them, so I'm not too stressed about this, but we also become close friends because they are also in Shanghai. So we obviously met up, and then we discussed a lot before their paper came out. And one of the good things is that they have done really hundreds and hundreds of different patients in different tumor types. And the key thing that they found is this one of these APC functions neutrophil, and more importantly, they found this VGFA positive and SPP1 positive subset, which correspond to our T3 neutrophil. So it's just to show that the power of using mouse model, while it may not be like a human study, which is really can be applicable to clinical setting, but however, with the mouse system, it can kind of explain, how it actually mechanistically, how this cell derived, how this cell got reprogrammed. So by combining all this study together, it kind of gives us a very good pictures of, like when we want to do targeting of neutrophil within this for treatment, perhaps you have to consider it like targeting it within the leukocyte or the in a tumor site, instead of from this using a systemic approach. So all these things will be kind of help us to better design if there's one treatment that can be like a neutrophil based kind of a treatment for cancer. So finally, I just want to spend like several minutes just to really talk about how we think about neutrophil, which can be applicable not just to cancer setting, but it can be in general, like how neutrophil responses can be modulated. So we know that now neutrophil have this very strong adaptation ability, even under resting states, that they follow this trajectory of development from I should say from pre immature to mature neutrophil, then release the blood, and then go to the tissue even at the resting state. And then Andrea's cell paper showed that even under resting state, when these neutrophil get into the tissue, somehow they have these adaptations, and they will become, they acquire these tissue signatures within, depending on which tissue these neutrophil are localized. And this is kind of easy to understand under resting state. But then under stress condition, then it becomes very complicated. Because as I mentioned before, under the stress condition, for instance, when there's inflammation, when there's a tumor, both the mature and immature cell can be released to the circulation and can be recruited to this leukocyte. And then this kind of raises, but one key point that I have to point out is that even though there is this expansion of neutrophil or in the bone marrow, but it is known that they do not kind of skip one step. They always still have to follow through this trajectory from the pre-immature mature neutrophil, that they would not kind of skip one step and become a mature neutrophil immediately. And more importantly, that one thing that we have been talking about, there's heterogeneity. But if there's a heterogeneity within these neutrophil populations, if there's a response, like for example, there's an injury that recruited all these neutrophil. So what does it mean? So it doesn't really make sense. If you can imagine, if each of these neutrophil is doing function A, B, C, D, E, F, G, then you may not have this productive effective function. Because for T cell, as we all know, that how T cell kind of carry out their function, because it's through clonal expansion, you will build out enough number to carry out a single function. Whereas neutrophil, they do not proliferate when they get into the peripheral site. So what they relies on is to recruit more and more neutrophil coming to this leukocyte. So what does it mean, like, if each of these are so different, including the maturation status as well? So what we believe is, at least from our tumor study, while this is so-called like the dark side of it, and we are now doing more experiment to show that in the protective response, perhaps that this uncoupling of this maturation program when they get into the tissue, when they get into the leukocyte, and these leukocyte or this local tissue will kind of reprogram them to achieve this at least the functional homogeneity, to ensure that they perform a single function at this leukocyte. And if you put all these things together, I have no answer, but this is just putting out there. Hopefully, we all can talk about it more, think about it a little bit more. So there are a few things that we have to consider when we look at our neutrophil responses. So as I mentioned, we need to think about the tissue compartments. And then from Peter, at the time, this Nature Com paper, they proposed that this is neutral time in which the trajectory never change. However, somehow, when there is this so-called the priming, or when there's a stress conditions, this kind of deviate out to have this special kind of a program, and then this will then relate to their function. So this is something that we should start to think about multiple things, and perhaps as we go, maybe there will be even more boxes that we have to consider. And finally, I have to thank all these lab members that are in Singapore. So this is a Singapore lab. I will always indebted to them. So in a way that without them, because all of them have been with me at least 7 years. So it's a long time with them. This group of good people that helped me to build my career, and very unfortunate, only one really brave soul went to I should say went to Shanghai with me. The rest are still in Singapore. And the good thing about when I say good thing is that my lab is kind of one split into three, because three of my postdoc over these past 2 years, they became independent. So so basically the lab member can absorb into these three different lab in Singapore, which is a great thing. And all the SIGN collaborator that all these PI that I collaborate closely with. And I always have to say this, it's very unfortunate all of us are not there anymore. So it can't say so much about our previous institute. So and I want to thank all these collaborators and all the real doctors in Shanghai that help us a lot. And finally, this is my lab in Shanghai, and we are always recruiting. Yeah, thank you so much. Yeah.
Are there any questions in the room? I've got microphones.
Hi, great talk. I have a question about, so these atypical neutrophils that you briefly talked about, are those the VEGF-A, SPP1 positive neutrophils, are they atypical neutrophils? And
They are what? I can't hear you very
so
clearly.
you talked about atypical neutrophils,
Yeah.
which is cyclic F positive neutrophils. Are these VEGF-A positive, SPP1 positive? Are these cyclic F positive neutrophils? And also, is there a way to in vitro transform the transform neutrophils into from your mature neutrophils to T3 or the immature neutrophils to T3
Yeah,
neutrophils?
yeah. So it's a good question. So the atypical neutrophil is I think in my earlier slide I talked about it is not the T3. So cyclic, I know that I think where you're coming from because there are some some tumor model neutrophil became cyclic F positive, but these are two separate things. So we do see the cyclic F in our T3 neutrophil, and why we did not kind of pick this cyclic F as the best marker at the time is that they do also express in other tissue when the mouse are tumor bearing. So we still use DC3R1. We think is a better marker. So regarding the whether we can transform this neutrophil, we can do that. So if you we have done this. So essentially we can use this tumor condition media, the cell line condition media, and we culture any neutrophil either from bone marrow, spleen, mature or immature, they can become T3 overnight after overnight culture. Yeah. So we have done like we culture and then we have done sequencing again, and then we map it back to our single cell data set. We have done whole series of this.
Is that based on the VEGF-A or SPP1 expression?
Here, can I get you some
Sure.
Is that based on the VEGF-A or SPP1 expression? What expression do you use to say?
So
like,
we use a DC3R1 as a surrogate marker. One the very first criteria is the DC3R1, and then also when we look at the single cell not single cell, we did like a nanostring, so maybe like 30 different genes. So we have a set and SPP1 VGFA are a part of these gene sets that we look at. Yeah.
Great work, thanks.
down front?
Hi, great talk. It sounds like the argument is that these T1 immature neutrophils gets kind of sidetracked to this pro-angiogenic function. I'm wondering, once they're in that state, do you think they continue to mature and then sort of exchange with that sort of population, or are they just forever going to be a different thing?
Sure. I think this is a fantastic question. We think about this all the time, and this is kind of coming back to this neutral time that we are talking about, is that when we put all these sequence, because this is kind of give you a trajectory kind of predictions. So when we put our T3 into this neutral time, it kind of show that perhaps this cell is already at this terminally differentiation stage. And when we say that, it's because of, for example, like if we take T1 and T2, if we isolate T1 and T2 from the tumor, we just put it on a culture like a normal media, DMEM, no stimulations, they will still become T3 after 1 day or 2 days. But whereas for the T3, we take it out, we put it in a DMEM, they will still remain as a T3. And so they don't switch back, they don't go forwards. So it's almost always like T3. And we did that also, like we take T1, T2, and T3, and we kind of transfer it back to the mouse, and we try to isolate it back, and they are always remain as a DC3R1 positive T3 neutrophil.
And the T3 will always stay partly mature and partly immature, or will it sort of age into a more mature?
Sure. So they over time they will be mainly become a mature neutrophil for sure. Yes. Yes.
So it looked like from your analysis, you have immature neutrophils going into that type of tumor. From your broad phenotypic analyses, have you looked at the adhesion molecules and chemokine receptors on these two populations?
Sure.
And might they be using the same ones or differentially?
Sure. We haven't to be, I have to admit that we haven't looked at it very deep enough, because I think at the time the paper, we kind of tried to write it in a way that it's not a tumor paper. We wrote it in a way it's like a neutrophil development paper, because we are so afraid that exactly that all these questions are related to tumor, whether we can somehow harness some of it as a treatment. So we never propose it as a tumor kind of a paper. Because like the very first thing people will always ask us, do you know what factors make this neutrophil become T3? We don't. We still don't know which factor it is. So these are the things that we are currently trying to work on. But it is a good question. We should look at whether the recruitment itself, whether it's changing or not. These are the things that we haven't. It's a shame that I moved because I just like when the paper is about 60% done, I move, and then we kind of try to complete this paper in Shanghai while some of my former postdoc that in Singapore, and all these small details that we couldn't really focusing on, and we should restart to look at it. But it's a good question. Yeah, we haven't done it. Sorry.
And just a quick follow-up. So I'm assuming you probably haven't looked at adhesion molecules on the vascular endothelial cells.
We haven't done that. Yeah, but I think these are all important questions that we should really try to address, since we have the single cell data set. We also have the spatial transcriptomic, and more importantly, we also have the protein staining that we should go back and look at all these things. Yeah, thanks.
Okay, let's take our last question down here.
Okay, that was a fantastic, very thought-provoking talk. Thank you. So I have one question about your data and then one philosophical question that you can just take with you and maybe we can chat later.
Sure.
So the question about the data is you mentioned just now that if you put the T1 and T2 in culture and you let them sit for a couple days, they become T3.
Yeah.
So when you look at the tumor data where the T3 were these pro-tumoral
Yeah.
VEGF secreting tumors, VEGF secreting cells, what happens if you actually mix T1 and T2 and put them in with a PDAC? Do you recapitulate the T3 effect?
So you mean that I mix T1 and T3 together?
T1 and T2 together, because the T3 looks like a mixture of T1 and T2, right?
Yeah, yeah.
So yeah, that result, can you recapitulate the T3 if you just mix T1 and T2 together,
Yeah.
as opposed to
So
separately?
we haven't done that, but if you look at them separately, if you really look at closely, in fact, they do have some sort of a anti-tumoral effect. So which is kind of a consistent with some of these data that are in a clinical setting that in which where they start to they found several not several, they found a population of neutrophil which can be anti-tumoral.
Yeah.
And APC kind of a neutrophil. So
But you have removed sort of the influence of the other cells in that system, right?
yeah,
So the interactions between
T1 and T2.
T1 and
So we
T2
haven't
might
done that.
actually drive a different environment than either one alone. I mean, I think the real power of spatial is that you can actually start integrating all of the components which behave differently together than in isolation. So I mean, the other thing that I think is really interesting is whether your trajectory of maturation, which seems sort of linear, is actually not more convoluted, and cells can go to T3 and then to T1 and T2.
So you mean like reverse?
Yeah, I mean, no, not necessarily that they're reversible, but that when you have the population together in the tumor, so T1s can become T3s in vitro and T2s can become T3s in vitro, but that in vivo the maturation is not that linear, right? That the three cells interacting together are doing something different. So I mean, I think adding T1 and T2 together, I realize these are not easy experiments, but it'd be a fascinating.
Yeah, we haven't done that.
Yeah. I think the other point is that it's fortuitous that mice and humans seem to have similar leukocyte distribution and function for the most part. But when you look evolutionarily, you know, the neutrophil compartments are really different. So in non-mammalian species, we only define them morphologically and they look
Yes.
different, right? They're heterophils or other types of cells. And then even in mammals, ruminants are neutropenic.
Okay.
So physiologically, they don't have a lot of neutrophils in the circulation. Elephants don't have a neutrophil. They have this bilobed thing that we don't really know what it is. So I think it would be really interesting to look sort of over the course of evolution how these cells have adapted and why, right?
Sure.
And why is it that mice look like humans, but you know, whales don't? I think there's all sorts of really interesting things about this neglected cell population that people don't pay attention to because they only live for 6 hours. So this was a wonderful talk. Thank you.
Yeah, thank you. I think regarding to your last comment, one of my colleague in Germany, he started to do this looking at evolutionary, what is the link, what is the function, why certain species or certain animal may or may not have it. I don't know the progress of this, but I think it will be a good thing to look at because in Guangzhou, there's this young investigator that he actually did his postdoc in the Edo Army's lab, and then he returned to China. So he started to look at this. He found this new population, not neutrophil, but more like a microglia type of cell in circulation. So he started to look at crocodile, donkey. I don't know, like 30 different things that I don't know how he get all these animals and how he gets the. It's amazing. So perhaps I should really talk to him to see whether I can look at some of these things that he has. But the problem with neutrophil, like you say, is like they die so quickly. But for him, it's much easier because like those sample can be frozen and he can still analyze it. But neutrophil is just not so easy. I have to go to the site because like he found this interesting place that every November they have to eat donkey in northern part of China. So he have to wait until November so that he can get some supply of this sample. Yeah. But so maybe I should go there just to do this experiment.
All right, well, we are definitely out of time. Thank you so much
Thank you
for
so
a
much.
great talk.
Yeah, thank you.